Make sure you learn what a hematocrit is, if you don't know already. It was on slide number 2 on this PowerPoint. I skipped it. I want you to read about a hematocrit on your own. Hematocrit. And look, I have some slides on here for the anemias, even though it's in the homeostatic imbalances, so you can use the slides to study as well as looking at your book for that. I will talk about this. This is sickle cell. Sickle cell anemia makes a red blood cell get this shape. Well, that doesn't look good to me. Does it to you? That's a great slide. It's malformed hemoglobin, okay, that causes this. It's actually the hemoglobin inside the red blood cell that causes it to develop this shape. Okay, tell me, what's hemoglobin? Okay, it's a polypeptide group, right? 4 polypeptide chains. It's a protein. In the protein, hemoglobin, this is crazy. A protein is just a chain of amino acids, right? Okay, it's 146. You don't have to know that. It's 146 amino acids long. The only difference between normal hemoglobin and sickle cell hemoglobin is 1 amino acid in the whole chain. It's the 6th amino acid. You don't need to know that either. But that's it. 1 amino acid is different. It's changed. It's mutated. The code, the DNA, you might remember transcription and translation and DNA replication and all that crazy stuff. Well, I know that it comes from areas where malaria is high, and the sickle cell gene actually protects you from malaria, which people that have sickle cell will get malaria and they'll fight it and they won't die from it. So their lifespan is lengthened, but it's shorter than the average person because it's down to 30. But why that? It's genetic. That's why. And probably because those peoples come from that area where malaria was. But do I understand it all? No, not all of it. Is that not crazy, guys? 1 amino acid on the chain is different. And it changes your life completely. And that's only 1 protein in the whole body. 1 amino acid on 1 protein, well, that's in 1/3 of all your cells, so it's pretty darn important. And the average lifespan of somebody with sickle cell is about 30. Not right now. They can't cure it. They're going to be getting better at stuff like that, at all the genetic stuff in the future. And it's going to get scary, actually. It's going to be cool, and it's going to be interesting, and you're going to be able to order up your baby with all the traits that you want. And one of those traits could be, I don't want my baby to have the sickle cell gene, so it won't die at 30. Then the other one will be, I want my baby to have blue eyes. So, you know, boy, I want my baby to be 6'2", not 5'2". You're going to have all these choices. Wait till I'm 100 and come talk to me, because I'll still be alive, because I'm going to have choices, and I'm going to grow a new heart. White blood cells, technically called leukocytes. Leuko means white. Look at that. Look how much of the total blood volume they make up. Is that not hilarious? You can't live without them. But they make up that little bitty amount of your total blood volume. We already know that. The count, what's an average count? 5 to 9,000. You could say 4 to 11. It depends on where exactly you grab it. But notice, the count's in the thousands, right? It's in the thousands. And the red cell count's in the millions. Wow, that's crazy. See, this is so cool. The white blood cells leave, they actually leave and squeeze through a capillary. That process is called diaphoresis, when they squeeze out of the capillary. Then they get out there into the tissues, There's a capillary. Here's what happens. When the white blood cells are rolling through a blood vessel, what they do is they will attach, and it would be, but anyway, it would attach to the edge here, and it would slow down. And it would slow down, and then it would go and squeeze through, and it would be out here in the tissues. So that process of squeezing through, what do we call it? Diapedesis. Once it's out here, it moves like an amoeba. You know what an amoeba is? It's a unicellular organism, kind of like a blob. So there you can see, we say that its motion is called amoeboid motion. Amoeboid motion. That means, and this is so neat, guys, it changes its shape as it crawls. It'll reach a little arm out, and that'll pull the rest of the cell. And then it'll reach an arm out on the other side, and it'll just be crawling along, reaching these little arms out. That's what your white blood cells do. And where do they go? See, if here's the infection, look at this. What a white blood cell will do is it's smart. It's like a bloodhound. It'll get a little scent, a little chemical scent, and it will move around until it finds more and more and more and more, and it will zero in on the infectious material that way. They call that chemotaxis. Specifically, they call it positive chemotaxis. But chemotaxis, what's that mean? It's like a chemical taxi. It's picking up a chemical scent, and it's following it. Because white blood cells, they take particles in to themselves through the membrane. They're just fascinating cells. You know, your white blood cells, they go places on purpose. Wrap your brain around that. Your little white blood cells, of which you are not aware, of which there are about 8,000 in every cubic millimeter of your blood, are going out into your tissues all the time and crawling around and looking for foreign material to either eat or destroy in some way. That is so, so cool. If your white cell count goes over 11,000, we would call that a leukocytosis. That is an increased white blood cell count. Common when you have an infection. That's why they take your blood when you go to the doctor. And they might do a differential cell count and see if it's eosinophils that are high, because if those are high, you might be infected with a parasite. Or if your neutrophils are highest, if they're like above 70%, your neutrophils are 75%, you might have a bacterial infection. Oh, wow. You mean that's how it works? What if your monocytes are high? Monocytes become macrophages. They respond to tissue damage, so you have some kind of tissue damage going on. Or what if your lymphocytes are high? Well, you got some kind of specific immunity going on. So there's all kinds of really cool things that they can determine through blood tests. So, you know, it's not really a waste of time for your doctor to take blood. In fact, they probably don't take blood enough, to be honest. And they just say, oh, it looks like this, so we'll do this. Here's the breakdown. We kind of know this stuff already. So I'm not going to spend time on it. I'm not going to talk about them again. I will say this, what we haven't said. Your actual white blood cells are divided into two categories. Granulocytes, which have granules, and you know for sure eosinophils and basophils have those. But so do neutrophils. Even though they're pale, they had little granules. I don't know if you were aware or not, but they did. Now, these cells usually don't live that long compared to red blood cells. But check this out. And I'm going to skip all of those because we've done all of that. There's their pictures. Those aren't good. Agranulocytes. Lymphocytes and monocytes have no visible granules. Kidney, spherical, or even a horseshoe-shaped nucleus on some monocytes. But here's the cool thing. They live longer, usually, than red blood cells. Macrophages live for months, some of them years. Lymphocytes can live for decades. That's crazy. But how long do nerve cells live? Your whole life. So, not so bad. Hopefully. Matthew. All right. Well, you mentioned that thing about alcohol earlier. It kills cells. Alcohol, that bad chemical. Hey, here's one thing. Oh, we'll let that go. Okay, so basophils, which I haven't mentioned, basophils contain a chemical called histamine. Do you guys know about histamine? What do you know about histamine? Inflammation. Histamine goes with allergies. Basophils cause your allergic symptoms. They cause it. It's a signal gone haywire. Late in the semester, or when we do the immune system, we'll talk about how antibodies play a role in that as well. And it's pretty fascinating, realistically. Anyway, histamine causes an increase in capillary permeability. You guys know what that means? Look, I'll use the same capillary here. It means it allows fluid to leave. And when fluid leaves, it swells up that area. Also, the blood vessels in that area, they dilate. By the way, Texans, it's not dilate. They dilate. No, they just dilate. Okay. That's what histamine does. That's why when you have an allergic reaction, you get all the fluid build up, you get the clogged stuff. It's not just histamines. It can be leukotrienes and prostaglandins and all kinds of stuff. There are another group of cells that are out in the tissues called mast cells, and they're basically like basophils that are in the tissues. They're different because basophils can be in the tissues too, but they're very similar. They do the histamine thing. They do inflammation. And usually when we talk about basophils, we'll talk about mast cells, and I'll say that exact same thing every time. So also associate histamine with mast cells. That's a great monocyte. Remember, know your counts on the white blood cell. Okay. Here's your white blood cell picture. Make it bigger for you. All blood cells are coming from what kind of cell? Hemocytoblast, which is a blood stem cell. Look at the path, though. There's two primary pathways and then multiple tiny pathways that determine which kind of white blood cell it will be. I do not care that you memorize these names. You know the first one and you know all the last ones, except for when we show the platelet later. I'll give you the name of the cell that platelets come off of. Leukopenia is a low white blood cell count. Hey, commonly medications can do that. Chemo does that. Leukemia is cancer of the bone marrow because it produces way, way too many white blood cells. I mean, we can be talking like 75,000 on a white cell count instead of 7,000. That's 10 times as much. I would like for you guys, in addition to reading about anemias, to read about leukemia. And then we get to the little old guys, the little platelets. They are fragments of this cell right here. Hemocytoblast becomes eventually a megakaryocyte. megakaryocyte. You do need that name. Look, it's big. That's why it's called mega. Karyo means? It means nucleus. It's a big nucleated cell. Oh, what a shock. It's not like a red cell. It didn't give up its nucleus. It didn't shrink down. But look here. This is what it's showing you. Remember how I said like the stamps? As fluid flows by, it just kind of peels little pieces off. They're little chemical sacs called platelets. Now, the books all say, and this blows me away, they say a platelet is 1/4 of the size of a red blood cell. We saw blood. Did any platelet look anywhere near 1/4 of the size of a red blood cell? I personally don't believe them. Maybe when a platelet's out in the tissues and it swells up and becomes sticky, but I don't, they're small. You see blood slides all the time. They're tiny. They look like 1/10 or 1/20 in my eye of a red blood cell. Okay? So now we get to the fun part. This is what I'm going to give you for your listening pleasure. Hemostasis. Hemostasis means the slowing down of blood flow. That is not the same, guys, as clotting. Clotting is only one of the 3 processes that stops blood flow. It's the big one. It's the one that really puts a stop to it, but it is one of the 3. See, first what happens when you get injured is your blood vessels actually contract. They spasm, they constrict, and that decreases the blood flowing through them. That's to help protect you. Anybody see Black Hawk Down? I know it was a long time ago. You remember that? When the guy was cut real bad by the leg and they were trying to get his artery, and they said, "It's running away from me, it's running away from me." That's what they do. The muscle in the vessels would constrict, and the blood vessel would pull up in the body because it's not just constricting in diameter, it constricts in length as well to try to stop the blood flow. But of course, if it's a big artery, you can't stop. It's not enough to do that. Then you get platelets clogging up the area and aggregating together. There's all kinds of chemical messengers that when you listen to this, you'll pick up. You need to know the names of things like thromboxane and serotonin. So when you hear that on the talk, make sure that you pay attention to that, okay? So platelets clog up. Tiny blood vessels, platelets can clog them up and actually stop some bleeding. However, in the larger ones, we actually need a clot to form. And platelets and red blood cells and white blood cells will all get trapped in that clot. And as we know, that clot's made of what? Fiber. Fibrin. And there is a whole pathway. There's a lot to this. And I'm putting a lot on you by making you learn this. But I kind of want you to. And you're going to see a picture like this. I will tell you in my talk, I tell you what you need versus don't need. Okay? I'm very particular. You don't need the names of all these numbers. There's very specific stuff you need, like intrinsic and extrinsic pathway and prothrombin activator, and the fact that calcium and vitamin K are clotting factors, very important clotting factors. And this slide will be very crucial to your understanding, and you'll hear me talk about it. And it may not be this actual slide that I have up there on the one that you're going to listen, but I will definitely go through this with you in that integrity. That's a clot. That's fibrin trapping red blood cells. Wow. And some platelets. You see them all over the place. This is a little big one and some littler ones. Looks like a spider web, doesn't it? Kind of a real thick spider web. And what's really cool is in the platelets, well, there's like actin and myosin, and they start to actually contract and squeeze and pull the fibers, and it squeezes fluid out of it and makes it stronger and tighter, which is really, really cool stuff. Yeah, see, clot retraction. All this will be on there. Okay, so what stops a clot from growing? First off, when you have a blood clot develop, or when you clot to stop that, you don't need it to stay there forever because the tissue is going to heal itself. So notice this. There's a process called fibrinolysis. We know words. What's fibrinolysis mean? Breakdown of fibrin. It begins within a couple of days. Who would have guessed that there's an inactive blood protein, right? That's what ogen means, that becomes activated through a certain mechanism that you don't really need to know the whole mechanism. But plasminogen is converted into plasmin, and it starts to bust up the fibrin and break up the clot. Why does it not happen instantly? Your tissue's not repaired, you would start bleeding again. So it gives you a couple of days head start on the repair so you can get some connective tissue in there, heal the stuff up, and then slowly start to digest the clot at the same time. It's fascinating the way that it works. Two things right here that prevent clots from becoming too large. Well, we have blood still coursing through that area. So the fluid that comes in there removes clotting factors and it dilutes them. That's cool. And then certain clotting factors are inhibited. And I really don't get all heavy into the names of clotting factors on there. So you don't have to worry. Blood clotting can be hugely complex and complicated, and the way that I talk about it is I usually stick with the basics. All right, there's this very important chemical that you'll hear about later on. It's called thrombin. Thrombin is what activates the fibrinogen. So thrombin is an enzyme that activates fibrinogen and causes it to produce fibrin. Therefore, some of the things that inhibit clot formation are called antithrombins. They would block thrombin. See, thrombin leads to fibrin, so if you don't want fibrin, you block thrombin. I bet 3/4 of you, at least half of you have heard of this. Raise your hand if you've heard of that. Oh, well, 1/3. Okay, good enough. Oh, maybe more. Okay. Heparin. Is heparin used in a hospital? Heparin is an antithrombin. It works against thrombin. It inactivates thrombin. So what happens if you inactivate thrombin? You do not produce what? The clot, the fibers, the fibrin. Oh, and I don't know why that's there twice, so my bad. Not big on that slide. There's the one. Okay. In a blood vessel, a clot is technically known as a thrombus. That is a clot that forms within a vessel. It is attached to the vessel wall. Can clots break free? And when they break free and they are free-floating, they are called an embolus. And as the blood vessels get smaller and smaller, a free-floating embolus would eventually get stuck in a small blood vessel, and then it would be called an embolism. So you don't, I hopped. Yeah, so it's not spelled there, so I'll spell it for you. I will say this. A, so you can see how I would spell this, cerebral embolism, there's embolism, can cause strokes. An embolism is what? That's an embolus. An embolism is one that has been lodged. Okay, so it was a thrombus, it broke free, it's an embolus, it lodges in something, and it's called an embolism. See, if we had a big blood vessel here, and there was a clot like this size in it, see, it could travel through these, and then as these branched off and got smaller, if it went this way, it could eventually get stuck right there. And then everything from this point down would do what? It would actually die if there wasn't collateral circulation, which there commonly is, but that's one cause of strokes. It's one cause of heart attacks. It's causes of lots of different things that go on. You cut off the blood flow, then you cut off the oxygen to all these tissues in that little localized area, and that area dies off. If it's a big blood vessel and a big clot, you got big problems. If it's a little bitty, teeny, tiny one, maybe not so many problems. Yes, sir. Oh, you're just stretching? Okay. Now, this is cool. Aspirin, how does it work? Aspirin inhibits prostaglandin formation. And one of the chemicals that you will learn about, which is produced by platelets, is called thromboxane. And it is a prostaglandin. Aspirin inhibits formation of that. That causes vasoconstriction, so you won't get that vasoconstriction going on. So it helps keep the blood vessels big. It also stimulates platelets to clump together and form the plug, so it blocks the platelet plug formation. Heparin we talked about. Produced by basophils and mast cells as well, just like histamine. And have you ever heard of warfarin or Coumadin? What's its other name? Yeah, they call it a blood thinner, right? Vitamin K. What do you know about vitamin K? It is considered a clotting factor. Do you know where vitamin K comes from? Baha, ha, ha. The bacteria in your colon make it for you. Without bacteria, we do not produce this clotting factor. We need them. One reason that antibiotics aren't always so good for you is they wipe out your normal flora, which makes a clotting factor right here, vitamin K. Coumadin or warfarin inhibits that clotting factor, so it helps to keep the blood thin. When they say a blood thinner, they mean blood that won't clot. Why is vitamin K important? It causes the liver, it stimulates the liver to make the clotting factors. So not only does the liver make plasma protein, your liver makes clotting factors. And the bacteria in your gut produce vitamin K that tells the liver to do that. Disseminated intravascular coagulation, what does that mean? Disseminated all over. Intravascular, within the blood vessel. Clotting, coagulation. These are blood clots all over the place inside your blood vessels. Wow. So what happens is then afterwards, your body's like blown up with clots, and then it loses the ability to make more clots, and then the people bleed. Bad news. Okay, just like I had you read about leukemias and anemias, I want you to just read about a thrombocytopenia. That means, what would it mean? Can you tell me? Right, penia means low, Thrombocyte means platelets. Did you? When you had the spleen problem? A normal platelet count, which is not on this PowerPoint, I believe, or maybe it was. I think it was back there. Do you remember what a normal platelet count is? See, this is crazy. Your book can't even, if you look on in your lab book, on one page it says one thing and on another page it's different. On one page it's like 150 to 400 and on the next page it's 250 to 500,000. The whole point, in my mind, white cells, thousands, usually less than 10. Red cells, millions, around 5. Platelets, 100,000s. Thousands, 100,000s, millions. There's huge differences there, right? Platelet count around 250,000 would be considered quite normal. How many functions of the liver have we already hinted at? Plasma proteins, clotting factors, bile, and gluconeogenesis and glycogenolysis. Glycogen storage, right, and breakdown and formation of new sugar from amino acids and fatty acids. We've already done 5 functions of the liver, and we haven't even got close to the digestive tract yet, or the digestive system. The liver has over 500 functions. It is crazy important to our life. We're going to talk about blood types. How many people in here know their blood type? Okay. Cool. How many people know how many blood types there are? The basic blood types are A, B, AB, and O. So there's 4 basic main primary blood types. And we have a couple of things to talk about with each blood type. The first thing that we're going to mention is a blood type is named for a surface marker that can be seen on the red blood cell. And the surface markers on the red blood cell are known as antigens. So, for example, this isn't what it really looks like. This is just how I draw it. There's a little red blood cell, and there's a little antigen, a little surface marker. We say antigen, you might be thinking receptor because we've talked about receptors on the surface of the cell, but they call these antigens. And this antigen is called the A antigen. So they call the blood type that has those, and it's not just one. There's lots of them on there, but they call that type A. It's really pretty basic. Type B, then, you want to take a wild guess. Just go out on a limb for me. Yes, it has the B antigen. It does not look like this in nature, but hey, I can draw a sideways B, and that makes everybody happy because you can see it. So this kind of looks like an A, and that kind of looks like a B, and we're all good. Now you got to really go out there and stretch your mind. What does type AB have? Why, it has both A and B antigens. And where are these antigens? On the surface of the red blood cell. They are part of the red blood cell. And then lastly, we have little type O. Anybody study ahead and already know about little type O blood? It has no antigens. As far as the AB blood types, the surface of this blood cell would be empty. Okay? Now, A's and B's aren't the only ones. There's like 30 other little minor ones. But we're not worried about those right now. We're just worried about your main primary blood type. The interesting thing about blood is that there's 2 parts. You have the cells and you have the... About blood, you have cells, formed elements, and you also have plasma. There is a portion, and I like to put, by antigens there, I like to put RBC. Just to remind everybody, that's on the red blood cell. Well, there's another part to each blood type called an antibody. Now, I know you associate antibodies right now with the immune system, probably, because we've talked about them just a little. However, blood also has antibodies in it. And the way that it works is this. Really, we're born with no antibodies. And depending upon what blood type you have, you will develop antibodies. You will develop the appropriate antibodies in your blood according to this formula. If you have, and I'm going to just draw right here and I'm going to erase it in a second, so just so you know, because I want this table to look nice. So don't draw this, whatever I'm putting right now, don't put it in your table that you're making. Okay? Do it somewhere else. You can write it, just do it somewhere else. If you have type A blood, that's an ugly red blood cell, but there it is. Here is an antibody for A. Do you see how the shape kind of fits right there? It's a bad drawing, so it's not exact, but hey, why not? Do you think that you want these things to stick to your red blood cells and cover them up? No. The answer is no. You do not want that to happen. Okay? Antibodies are made to stick or attach to the antigen and cover them up. And actually what that does is the way an antibody is, this isn't the real true shape of an antibody. We'll see those later on. But what the antibodies do then is they would cause other red blood cells to stick to that and other red blood cells to stick to that. Do you think that you want your blood to clump inside of your blood vessels? That sounds like a bad thing, right? It's different than clotting. It's clumping all up together. But we do not want that to happen. Therefore, naturally, if you are type A, would you develop the antibodies for A? That would make no sense, right? So what do you think type A people develop? Right, and they call them anti-B antibodies. The reason they call them anti-B is because they bind to B. So they're kind of against the type B blood. Well, by the same token, I'll draw this one. If somebody has cute little B blood right there, do you think that they want these things floating around in their blood to come over and attach to them? The answer is no. So what did they develop in their blood? Look, they develop the ones that look like that. They develop the anti-what? Anti-A. And we call it the anti-A. What word goes with it? Antibody. Antibody. Now, blood types are very easy to understand and to see. It takes some time because there's all these A words. And you got to get the A words straight and you got to figure out where they're at. So let me recap for you. 4 basic blood types. Each blood type is named for the blank on the surface of the red blood cell. Antigen, named for the antigen on the surface of the red blood cell. When we're young, we develop antibodies to our own blood type or not? No, to the opposite. Since the only 2 antigens that we're talking about now are A and B, if you have A, you develop the antibody for B. If you have B, you develop the antibody for A. You want to take a wild guess what you develop if you have both of these on there? None. Good answer. Those people would have no antibodies. Why would they have no antibodies if this is your blood? Because this one would clump and this one would clump, right? Either of them would clump. So naturally occurring, you would not get those or those for type A, B blood. And then last but not least, the little O people. Raise your hand if you're a little O person. Wait, where are you sitting? I want to see this. Raise them up. Look, look. Oh, you ruined it. See, they were all together. You're supposed to be sitting with them. Okay. Type O has naked cells, so they can have this floating around, and they can have this floating around, and it's got nothing to stick to. Do you guys see that? Type O, therefore, has what? Both the anti-A and anti-B antibodies. And that's blood types. That's easy. That's the easy part. Naming the blood type is the easy part. I'll probably ask you for one of the blood types. I'll just pick one, and I'll say completely describe a blood type. Oh, who knows? I might pick all four. Say completely describe all four blood types. I won't tell me the antigen and antibody, because I don't want to give you those words. I expect you to know if I say describe all four blood types, you should do something like this. And then the fun part is the next page. We need to see about blood donating. We need to know, we need to be able to see which person can give to which other person based on blood type. So when we do this, we're going to look at a very specific formula. We are going to look at the donor, and when we look at the donor, we are only going to be concerned with the red blood cells. We're going to ignore the plasma of the donor for now. We're not talking about donating plasma. We're talking about donating blood, which gives the blood cells, which carry oxygen. And on the person that receives the blood, what would we call that person? Yep, recipient. You want to take a wild guess what part of their blood we're going to look at? It is their plasma that is important. Because you have a lot of plasma, a lot more than they'll be giving you in the blood. So you have a lot of antibodies that are already there in your blood. You don't want to receive any cells into your blood that your naturally occurring antibodies would attack and clump. And that's the formula. See, as the person that receives the blood, you need to make sure that you don't get the wrong red blood cells in there. That's the important key. Right? When they're donating plasma, they mess with the plasma. They can take out the antibodies if need be. There's not going to be enough of them in that blood to clump your blood together enough to make any kind of big deal. So this is the formula that we look at. The donor's red blood cells and the recipient's plasma. What is on the donor's red blood cells? Good. Antigens. And what is in the recipient's plasma? Antibodies. So here's what we're going to do. We're going to just work it out and see what happens. If a donor is type A, I go about figuring this out in a kind of backward fashion. I want you to understand this. This person cannot donate to people that have what floating in their plasma? Anti-A, correct? They cannot give to the anti-A people. Flash back to your chart. Who has anti-A in their blood? Type B. Who else? Type O, right? Remember, in the plasma, type O has both, okay? And type B has anti-A. So we know they can't give to anybody with anti-A. So they cannot give. And this part, notice how I'm pushing it off to the side. This isn't what I usually ask you. I don't say, who can they not give to? But that's the way you figure it out. We know that they can't give to B or O. So who does that leave that they can donate to? A and AB. Do you see that? Would it not make sense that you could give to your own type? That kind of makes sense. Okay? And type AB blood has empty plasma. And if it has empty plasma, you can put whatever cells you want in there. So by that token, what do you want to guess about type AB? It's the universal recipient. We're going to see type AB on all these. Okay. Let's do type B. Type B can give to anyone that does not have what? Well, let me rephrase that. They can't give to people that have what? Anti-B. Who has anti-B? Type A and type O. I know it takes time to work with this before you can just spit the answer right out. Right now, you're probably having to look at your table. But on the test, you can draw your table out, and then you can look at it and figure it out. That means that type B can give to itself and AB. Are you seeing a pattern here? You can donate blood to your own type and AB. Typically, that's how it works. So by that formula that I just gave you, you can give to your own type and AB. Who can AB give to? Well, that would only be AB. They can only give to AB. Why? They have both antigens on the surface. So if there is anti-A, it would clump. If there is anti-B, it would clump. They can't give to anybody that has those things. Well, everybody has them except other ABs. AB can only give to AB. Where's my type O people again? There we are. By the way, I'm one of these people. Type O has naked cells, correct? They're little empty cells. You can put those cells in any blood because there's nothing for them to stick to, those antibodies to stick to. So therefore, type O can give to any blood type. Type O is known as the universal what? Correct, the universal donor. So they can give to A, B, AB, and O. Universal donor. I want you to know that type AB is the universal recipient. So anyway, RH factor. And yes, there's some PowerPoint information on this. Here's what we just did. Okay, notice anti-A and anti-B, the antibodies form in the blood around 2 months of age or so. It's not an exact perfect. The RH factor, or RH blood groups, different. Instead of naming them, because there's, wow, look at that, 45 different RH factors. We're just going to say a person is either RH positive or RH negative. You know what RH positive means? It means they have the RH antigen, one of them, or multiple ones. They have RH antigen. You know what RH negative means? No RH antigen. It's all about the antigens. Where are antigens found? Excellent, on the surface of red blood cells. Therefore, many of you already knew this and you were going, I know my blood type, it's O positive. And you were wondering, well, where's the positive part come in? That means you also have RH factor as well. Okay? Now here's the cool thing. Naturally, none of us have the antibodies. It doesn't matter. This one's different than type A and B. You do not develop the opposite antibodies with this. The only way that you develop the antibodies is if you get Rh antigens somehow into an Rh negative person's blood. Because an Rh negative person has no antigens, so they will develop the antibodies to fight that, if that kind of blood is put in them. Okay? So if a Rh negative person is given Rh positive blood in a transfusion, they would develop the antibodies and clump it. All right? But just in their normal life, odds are they're not going to get that blood in them. If an Rh negative person and an Rh positive person decide to become blood brothers and they cut their hands and they shake their hands, guess what the Rh negative person is going to all of a sudden start to do? Build the antibodies. They're not going to get enough blood in there to make a big difference. They're going to have these antibodies. Are those antibodies going to affect the Rh negative person's normal blood? Nope, they're just going to be there. Okay? It's really no big deal. Except when? Ladies. Except during pregnancy. So here's the deal. You know what that is. I drew it before. Okay. He's got a little bow tie on. All right. So there's the little baby. It's a sad little baby. And this is the mom. And we are going to say that the mom, for right now, just go with me. We're going to draw this multiple times. If the mom is Rh positive, is there any situation that would cause her to develop the antibodies to this, to the Rh factor, if she's positive? If you're Rh positive, would you develop antibodies to kill your own blood, to destroy your own blood? So is there any way a positive person could develop them? No. So we never worry about a positive mom. Okay? If there's a positive, Rh positive mom, this doesn't even matter at all. Okay? It's only if the mom is Rh negative that we have anything to talk about here. And if the mom is negative and the little baby that is developing is positive, see, we have this temporary endocrine organ that I draw a little funny looking thing like this. It's called a placenta, right? And what does a placenta do? Mom's blood vessels come in here, baby's blood vessels come in here. There's a little space between them where stuff can diffuse back and forth if it's small enough, like oxygen, like sugar, right? Like electrolytes. It's made to do that. Stuff can move. Well, if because things are little and stuff happens, some of baby's little blood gets up here, what would mom start to do to those Rh positive antigens? She would start to make antibodies. Here's the good news. It takes a long time. It takes months for her body to do that. It took months for the baby's blood to develop to the point to have those. So by the time the first baby is born, well, I said it wrong, I guess. The first baby is born before the mom develops the antibodies enough to harm the baby. So the first baby is always okay with this. However, baby number 2, baby number 3, baby number 4, baby number 500. Wow. That's a big uterus. Okay. Each subsequent baby, if they are Rh positive, then the mom already has these. Her body has the memory of that. As soon as the baby's blood gets up there, she cranks up her production really fast, and it harms whatever positive baby this is. Okay? Once again, our formula is the mom has to be Rh what? The mom has to be negative, and the baby has to be the second baby that's, or more, that's Rh positive, right? What if the first baby was negative? Then it wouldn't produce it. So the second baby that was a positive baby is really the first positive baby. You see what I'm saying? So it's the second positive baby, and third positive or fourth positive or whatever. Any other negative kids that come along, they wouldn't be affected by this. So what does it do? Well, it clumps the baby's blood. The red blood cells clump together, just like we said. That causes more of them to be destroyed. And when you're destroying more cells, what does the body do to compensate? It produces more. So you have two things going on, excessive destruction and production of red blood cells in the baby. So they call this disease two things. They look at it two ways. Erythroblastosis fetalis. That means increased red blood cell in the developing baby, right? Erythroblastosis, increased production in a baby. They also call it hemolytic disease of the newborn. Why hemolytic disease? Hemolytic. Blood destroyer, right? Red blood cell destruction, hemolysis. So they name it both ways. Now the good news, when the mom was negative and the baby was positive, it doesn't happen all the time. It only happens sometime. It's a low percentage of the time. But when it happens, these babies usually didn't survive very long. So Rh negative moms get to take a shot called RhoGAM to help inhibit the production of those antibodies. Okay? Rh positive mom never has to worry about that because she won't produce them because that's her own blood type. And she wouldn't produce them for her own. But if it's a negative mom with a positive baby, they give them that. It's a gamma globulin shot. And it blocks the antibody production. So that's pretty cool.